Okay, picking up kind of the building on genetics and kind of working it together with the evolution that we did last chapter, is that one of the things that supported evolution is being able to compare features, homology and analogy and that sort of thing. With genetics, we can compare that on a molecular level. If proteins are long sequences of amino acids and mutations can change that sequence, then you start with one protein and it goes up one branch of the family tree, changing here and there. It goes up another branch of the family tree, changing here and there. If you compare those two proteins, places that they are different will give you an idea of how long they've been accumulating differences. And the downside of that is every amino acid change isn't the equivalent of every other one, because some of them do mess with what the proteins do, and if it changes the protein in a bad way, it's going to be, those possessors are probably not going to survive. But they used to compare proteins. Now they compare DNA sequences. A huge amount of our DNA isn't actually code for anything. It's doing different things, but it's pretty changeable without really messing it up. So again, you've got a stretch of that kind of DNA in your ancestor. But the two groups split. One's going to this species, one's going to that species. They're no longer recombining their genes. We'll get to that in a minute. And they're accumulating mutations in that sequence of DNA. Different mutations, just random. The longer those two branches have been separate, the more different mutations there are in each group. So that's your molecular clock, is that it's like, how often do the mutations happen in each thing? Well, every 10 generations, every 15 generations. It's not that easy to determine. Uniformitarianism, we assume that these mutations have always accumulated at the same rate, but who knows? Conditions in the past may have changed mutational rates. But it's a way to compare different groups by looking at how different their DNA stretches are. And it's like every mutation is a tick in the clock. People started to mess around in the lab with trying to induce evolution in things that reproduce pretty quickly. You change conditions and see what happens in things that reproduce in the lab pretty quickly, right? Conditions drive evolution. You can see it happen. There's many, many, many examples of that. Well, sometimes you would change conditions and there would be members in the group that were ready to go. They had some variation that was kind of preset to deal with these changes in conditions. Now, Lamarck, the guy who thought it was all a big plan, his idea was you get what you get when you get what you need when you need it. And it kind of sort of looked like that was happening. And people went, well, no, these are genetic mutations that have happened a while ago, and they've just been carrying on through the population, and now the conditions are giving that an advantage, but it's been there. And other people went, well, one mutation would have died, just faded away, right? If it wasn't really helping anything, wouldn't it have just faded away over time? Math guys, Hardy and Weinberg, math guys. And their rule was, okay, you have a new allele, you have a new mutation. It won't go away. In fact, as offspring with it are, produced generation after generation, you may get more and more individuals with it, but it'll stay about the same proportion in the population. There isn't any reason for it to disappear, as long as these things are true. It can't be bad. Right? If there is some survival disadvantage to it, then people are right. It'll probably be less and less common as the survivors don't tend to have it. It also can't give them a disadvantage in reproducing, sexual selection, right? Survival, reproduction. And as long as it's not good or bad for that, there isn't any reason for it necessarily to get more common. You see, that was the problem with hybrid vigor. It's like, how did something like sickle cell become a decent fraction of certain populations? Well, these were populations exposed to malaria. But as long as there's no upside or downside to this new allele, maybe there's an upside down the road when conditions change, but in the meantime, as long as it's not being picked against, it'll hang around. You got a new allele through mutation. You can't come in and hit it with a stick again. Don't mutate it again, or it's not going to be there. But the thing is, remember, mutations are pretty rare. A mutation that changes function in some way, because it's not going to be an advantage down the road unless it changes something about function. You just can't change it again. But the odds of changing it again in a significant way are pretty low. So that's not, it sounds like a big deal, but it really isn't. What they were looking at is this allele's position in the population. Say it's 1% of the population. Population grows, 1% of the population is more individuals holding it. But if the population fragments, a subgroup goes off, it may take more possessors of that allele with it. And so now the fraction is bigger in this small off group than it was in the starting group. Maybe it doesn't take any of them, and now the fraction's bigger in the starting group. If an old group that has gone off comes back in and recombines, collects with the original group, that changes the numbers too. So this is a numbers thing, is that the whole idea, this is actually called Hardy-Weinberg equilibrium. The fraction of the population that has this allele is going to stay the same so long as these things occur. Now I've got a few terms I'm going to toss in here at the end. You need a big population, because in a big population, you got a little population, and this weird allele is only held by 2-3 members of the same family, and something bad happens and wipes out that family. It may have nothing to do with that allele, but that allele is now gone. Whereas if it's 7 families now, because it's been spreading as the population got bigger and bigger, then the odds of some chancy circumstance taking that allele away gets less. It's the same thing you do when you running scientific experiments. You run lots and lots and lots of repetition. Because if you only do 1 or 2 tests, you never know what sort of weird stuff is going to happen that's going to give you results you can't rely on. You run 100 tests, and yeah, maybe a weird thing happens once or twice, but overall you tend to find out what's a reliable result in an experiment as well. Okay, there's some terms that go with this. But this was the list I just gave you. Now we're going to go and kind of throw some new terms in. This both has to do with a small fragment of the original population. In the founder effect, that small fragment migrates away and changes the proportions. In the bottleneck effect, the population itself comes very close to going extinct, so it becomes a very small fragment of the original population, and whatever mix of alleles is left in those survivors, they now become the beginning of the population that expands. Give you an example of founder effect. Excuse me, is that blood type A is fairly rare in Native Americans. Doesn't really have any natural selection advantage that anybody knows of. There's been some guesses, but it doesn't seem to. Doesn't have any reproductive advantage, no sexual selection. But it's just rarer in that population than in the rest of the European population that came from the other way. The Native American population dates back to East Asia. East Asians came across to the Americas, they became the beginnings, a migrant group became the beginnings of the Native Americans. But it wasn't a huge starting population. That little fraction that went from Asia over to North America and then eventually reproduced and spread across North and South America started as a fairly small group, probably working their way along the coast, possibly in small boats. So say it was just a couple hundred people. That's a very decent change from the hundreds of thousands or millions of people in East Asia. So A is a little bit rarer in East Asia than it is in Europe, so it already would have been a smaller fraction of the population. Say you've got 100 boats, mostly with families in them. Got a couple of families where type A is fairly common. On the way, one of those boats sinks. Whole family is lost. That fraction of A in that small population has now just plummeted. Maybe there's only one family left with A. And that founder population now gives rise to everybody else that we now see Native Americans in North and South America. So A, for whatever reason, started out rarer than in the Asian population, and that's the way it's continued. Hardy Weinberg said, no natural selection or sexual selection, none of this other stuff. Now the population's big again, but that's the founder effect. Bottleneck effect has to do with the population being almost wiped out, and then this little group now being the start of the population as it grows back out. It happens a lot of times. Extinction doesn't always wipe everything out. If you look at cheetahs, the genetics of cheetahs indicate that they might have been wiped down to maybe a family or two a couple 100,000 years ago, and there's still, issues in there. They're genetically a lot more similar than you would expect a large population to be, unless it started as a bunch of relatives. People. A few million years ago, people started in mid-southern Africa, spread out across the Middle East, spread across Asia, northern Africa, and then something happened. And those people found they're not there anymore, and they were gone. And a big change, looks like it wiped out all the people except maybe a population in southeastern Africa again. So you had a bottleneck effect. And then eventually those people spread out again across the world. But what if the groups that spread across the world in the first place had not been wiped out? They'd already been, probably adapting to the different circumstances already, and they would have gone on to be who knows what kind of population. You look at how different the populations of people are now. It's not a huge, it's mostly cosmetic differences, but would have been very different if that bottleneck effect hadn't affected people. Another thing that happens is mutations are random. And if you get a mutation and it produces a change that for one reason or another tends to become more common, then leopards start to look different from jaguars. They exist in the same niches. They were isolated by the Atlantic Ocean, but jaguars tend to be a little bit bigger, a little bit oranger. Their spots look different. Leopards are a little bit smaller, a little bit yellower. Their spots look different. What did that? Is there some advantage to being orange over yellow? Maybe. Or it could just be that they have genetically drifted away from one another. That there isn't, you can't always look at a trait and go, well, what was the advantage there compared to this over here? Sometimes things just drift because remember, mutations are random and selection isn't always that powerful a driver. And sometimes you select for an allele and a linked allele right next to it gets carried along for the ride, even though it doesn't really have a huge advantage to it. But things that are linked together tend to ride together. Once that group of East Asians was settled in North America, there wasn't a lot of back and forth going on. The migration was pretty separated. And once you now have two groups, they're going to evolve as separate populations. Reproductive isolation is a big driver of speciation, of new species from starting populations, from starting ancestors. And there's a number of different ways to produce isolation that's reproductive. The groups don't reproduce with one another anymore. There was a time during the ice ages where it was easier to get from Asia to North America and probably back again. But that period didn't last, and North America and East Asia got more isolated from one another. The ice and the other things, the rise of the oceans, is that they disconnected the two groups geographically. Areas dry out, become deserts, separate groups. Rivers get wider, separate small things that can't easily cross a wide river. Mountain ranges go up. There's all sorts of things. I mentioned before the Atlantic Ocean. There's a reason why the continents look like they fit together like a jigsaw puzzle. They were all connected to one another at one point. And then the oceans, the Atlantic Ocean is getting wider about as fast as your fingernails grow, making the ocean wider and wider and wider. And it separates groups from one another. They can no longer mix genes, and so they start to accumulate these random mutations, these random changes, and sometimes different conditions on these different parts geographically. Sometimes what will happen is a small group within a larger group will isolate out for different reasons. One of them is niche isolation. Remember, a niche is a job being done in the environment, some sort of role that a population is doing. So say you've got a big population of day hunting birds, basically hawks. But there's a small subgroup of them that has some advantages at dusk and dawn. They can see better in the low light, and there's less competition for food then. So they become much more active, less active in the middle of the day, more active in the dusk and the dawn. And now selection starts to pick better night vision, quieter flying, better hearing. And they're no longer active at the same time that the other hunters are. So in terms of access to mates, they're accessing mates in their particular niche. In this case, it's a time of day. And what you get eventually is owls, is animals that are now very well adapted to complete darkness. They didn't start that way. They started as day hunters, but they isolated themselves in a different job. The iguanas on the Galapagos Islands isolated themselves into different niches. There's all sorts of examples of that all over the place. Sometimes you get a group that for one reason or another doesn't do its reproduction at the same time as the rest of the group. This happens in plants a lot, where for whatever change you'll get like a stand of flowers that's flowering like a month after all the other ones have flowered. And now they're no longer mixing their genes. As one group's flowering in April, the other one's flowering in June. They're no longer mixing their genes together. And June and April are slightly different conditions. You'll see over time adaptations to, if they started in April, the June ones will start to be different in various ways. And that's temporal isolation, isolation by time. And it's generally time of reproduction. Now, in things that reproduce, animals where there is mating behavior, there's mate choice going on. Sometimes you'll get subgroups that prefer one type of behavior over another. You look at birds of paradise, is that related birds on islands, but they look pretty different. And the way they produce, they give gifts to the females. The gifts may be different. They may build bowers for the females. The bowers may be different. Is that it's the behavior that separates one group out from another group, and to the point where they become reproductively isolated and then go on to be different species. Later in the semester, we'll talk about insects. And on the books, there's more species of beetles than any other type of animal. But it literally is on the books, because people have been collecting insects as hobbies for a very, very long time. Beetles are very accessible. And there's like a premium to being able to name a new species, right? You get to name a new species, that's pretty cool. So these people collect these beetles and look for these minuscule differences. And all they need to do is convince their buddies that they've got a new species. And there's a little bit of postmodernism there, because the buddies are hoping later they'll do it for them. But what they have found, and I don't know that they would have found this any other way, except they're looking really closely at these things, is that sometimes you'll get a subgroup of beetles where the penis is shaped a particular way, and they can only reproduce with some of the females in the group. Is that they're mechanically incompatible with some females, but so you have a subgroup that can only reproduce because they can only fit together, and another subgroup that can only reproduce because they can only fit together. And the assumption is eventually they'll start to separate by genetic drift, but a lot of times there's no other real obvious difference between these subgroups. But the implication is they're gonna be different over time because they're no longer mixing their genes together. There's a lot of chemistry involved in reproduction. In flowers, the pollen has to land on the female part, and then the female part has to help the pollen build a tunnel down to where the sperm is going to go and meet the egg cells. If you get a female flower and the pollen from the right species lands on it, but the female isn't chemically compatible with it, it doesn't make the thing happen. If you have a subgroup of that, again, you've got a subgroup that can only reproduce within the subgroup of the big population. And eventually you get drift of various sorts and they become separate. In people, in animals, sometimes it's a matter of, you'll hear sometimes, they went to the the doctors, and the doctors told him that she was allergic to his sperm. I've heard people say that. Typically, you're allergic to some chemical in their semen. You produce an inflammation. The sperm can't work in the system the way that it's supposed to. You can't reproduce with that particular individual. That happens sometimes in subgroups of animals also. So this is literally reproductive isolation, because reproduction doesn't work in these two things. Here, choice is different. Here, fitting together is different. And here, just the two things don't mesh chemically properly. Okay, that is the end of, okay. Well, I'm going to leave a couple of things to the next one because this is getting too long, but the next one's going to be really short.